if you're not careful, you can like lose sight of the disease that you're trying to solve.
So for complex immune diseases like rheumatoid arthritis or Sjögren syndrome, you are studying, part of your work has been devoted to study these disease risk alleles and what's the molecular mechanism that is basically underpinning them. So why is this important? Why this offers opportunities in terms of treatment or pharmacological intervention?
Sure. So maybe there's two ways to talk about this question. One is in terms of autoimmunity, and then the other is maybe in more general terms. But it's roughly the same thing. For autoimmune diseases, most of what we know about the immune system and how it works comes from model systems. And a lot of the motivation to develop new medications is really motivated by the experiences scientists have looking in these models of disease. So, arthritis in mice, right? And some of these treatments have some of these approaches have really motivated great treatments, and some haven't. So I think in my thinking, it's really important for us to understand what causes disease from a human perspective, so that we can treat it. But the reality is, there's very limited ways in which we can study causal mechanisms in humans, right? With advanced data collection, we can collect all kinds of data on individuals. We can get data on the microbiome, all kinds of single cell data. But really, at some level, most of the things we're looking at, it's very difficult to know if they're driving the disease or if they're happening as a consequence of the disease. So one of the reasons I really like human genetics, and we do the work that we do, is that the discoveries that we make from genetics are the things that are causing a disease. The loci that we're finding for rheumatoid arthritis or Sjögren's, those are the places where there's small differences between individuals that end up actually leading to a disease. So that's the good part. The bad part is, as we've been doing this work over the last 20 years, what we've realized is most of these loci that we discover, trying to understand what they're doing functionally to cause disease, what they're doing at the molecular level, is actually quite difficult. So that's the space that we live in. And I talk about it for autoimmunity, but actually this could be said for neuropsychiatric disease. It could be said for all kinds of other human diseases.
Yeah, we're all in that endeavor of the mechanisms and how to recapitulate them. So related to this, what's your opinion on extending the data collection to other human cohorts? Let's say, because right now most of the biobanks that we have are basically to European ancestries or from European ancestries, but obviously disease risk alleles do not only happen in European ancestries, they happen in different ancestries with different mechanisms. So what is your stance? Do you think that when are we going to get more data from more diverse populations?
Yeah, I think it's really important, right? Because as you said, diseases affect multiple populations. I think we want to be confident that our discoveries are generalizable, right? And different populations have different environmental factors and different, genetic histories, genetic ancestry histories. So if we're seeing something consistently across multiple populations, it really increases the confidence that we know what we're doing and that we're coming to something that's causal. Having said that, it's absolutely true that most of the data out there is still European ancestry data. And some of this reflects the fact that certain countries are the countries where people are making the investment. Some of this reflects sort of behaviors in the field early on. Like when 20 years ago when we were starting to do human genetics, there was a tendency for people to say, let's try to keep the study simple and recruit in one population. And so then that population becomes the majority population in the US very quickly. And And so you end up in this situation where you have a lot of European origin individuals in the study. I know we in our center, we're making a huge effort in Boston to try to recruit non-European individuals so that we have representation, of something that looks like Boston. And I think it's also really important for us to start, building biobanks in countries outside of Western Europe and outside of North America. So it's happening. It's slow.
It's slow. But
and obviously, once it happens, there's the issue of procuring the data, sharing the data, which means, dealing with all these international issues.
Yeah, which is it's a real issue that is, geopolitical also.
Yes.
So, moving on, you've mentioned the genetic component, but also the environmental lifestyle, right? And for the broad audience, let's say, sometimes we get a lot of these messages that this disease has all of this genetic component, all of this genetic, let's say, inheritance, and sometimes people get the message that everything is very deterministic by genetics, let's say, but we know there's a huge lifestyle component, a huge environmental component to it that is different between different populations of humans. So, for the broad audience, let's say, when you study the genetic component of a disease, in what ways that helps also get the broad sense of what is happening with the disease? So how do you integrate that also with the lifestyle component? So just say, we know it's not everything determined in the genes or the SNPs at all for most of the complex diseases that make the majority of the burden on the health systems of Western societies, let's say. But how do you go from genetics to also integrate the environmental aspect of it? It's a complicated issue, we know, but how do you see that?
Yeah, it's a really tough question. I think part of why we like studying genetics is because we can study it, right? We all have the same genome, we can measure it, we can control for confounders, we can look for differences and risks that are associated with different genetics. Environmental elements are much harder to study, in my experience and my opinion, in part because we're not good at measuring the environment, right? If you want to call it the exposome or whatever you want to call it. For autoimmunity, there's a lot of interest in viruses and pathogens that cause autoimmune diseases. There's a lot of interest in pollutants. We know smoking, right, is a risk factor for autoimmune diseases, established risk factor, and that, of course, makes you start to think, what about other things that we're inhaling? Could they be getting us closer to disease? And of course, diet is something you have to think about. The trouble is all of these different variables are very correlated with each other, and trying to disentangle, drivers and passengers. As you can imagine, someone who's living in a highly polluted area, that environment is coming with a lot of things. It's not just the pollution. It may come with socioeconomic differences in diet, differences in the kind of work they do, the kind of household they live in, et cetera, et cetera, et cetera. The kind of person who smokes cigarettes may have a different diet, and trying to know which of these things is actually driving the disease is hard. So really the only way to do it is to do clinical trials to really understand the key environmental drivers. So I think it's a really tough area. It's very important, but
difficult.
But difficult to
And there's also a lot of reverse causality, right?
Yeah.
What you mentioned. Okay.
A lot of also data that you don't always have.
Yeah, the electronic health records that you work with they are incomplete or
they are incomplete, not always true.
Yes.
Outdated.
We all lie.
Well, look, people talk about the electronic health record like it's it is an amazing thing. But if you think about it, when you go to a doctor and you have your physical appointment, your annual physical, or even if you're going because you're being treated for a disease, your interaction with the doctor might be 15 minutes, 20 minutes, maybe 30 minutes if they've got a lot of time. That interaction gets documented, Who feels that they've really communicated everything that's important to their doctor in those 15, 20, or 30 minutes? Nobody feels that way. And then that content that the doctor gets, it's probably not properly transcribed into the medical record. So there's a lot of gaps between the reality of what's happening to a person and what's going into the medical record.
That's true. And so, like you were talking that in your work you combine a lot of different data, a lot of different aspects to understand the diseases. So there's of course the genetics, there's the exposome, the environment, there's immunology, and so on. And so when you put all these together, like what questions can you ask, can you understand, and really what can you learn from all these things together that you cannot do with a single field, so only with genetics or only with the environment, so only with the clinical trials and so on?
Yeah, I mean, I think for human work, I think in a way this is something we have to do, right? Because mostly what we can do with humans is measure things. And so one of the things I think we try really hard to do, because more often than not we can't kind of perturb the system, right? So what we do like to do, and I think is very important, is like look for consistent signals across multiple data types and multiple modalities. So if the genetics tells you, this particular cell state drives disease, okay, that's very interesting. But now you need to look in the tissue and see what's there. So you've looked at the DNA, but now you should look in the tissue and see, okay, is this particular cell state, is it showing up where I think it should show up? Then you might be interested in the environment. And then you need to ask, okay, we think this environmental factor is playing some sort of role, in driving this particular T cell population. Is there actual evidence of that at the molecular level? So I think sometimes we're doing it in a holistic way, but sometimes we're just trying to do it in an algorithmic way. I think it's, to get to something that reflects, useful, actionable biology, we need to find something that we're seeing, across all of these different modalities all at the same time.
So, moving on to something more concrete, let's talk about your work on CD4 cells, CD4 T cells, and fibroblast on rheumatoid arthritis, because it's also related to this, right? In the past, let's say, we all access blood because it's a readily available tissue. Blood work it's the first thing that you get done when you go into a hospital for whatever condition. But now you're starting to tap into the stromal component of certain autoimmune diseases, right?
Yep.
That has always been, let's say, concealed, because obviously you have to go through some type of more invasive biopsy or some type of tissue extraction. But what are we learning from this, let's say, more stromal plus the immune component in autoimmune diseases that was not there before, when we couldn't access to all of these different cell types that are involved, not only the classical work on immune components and CD4 T cells, but also looking at the stromal part of the disease. What are we gleaning from that?
Well, I think we're learning a lot of things, but I'll try to give the high level points. I think one thing that's been surprising to me, because I started as a geneticist, and like many geneticists who are interested in the immune system, I started by looking at immune cells in the blood, and of course, there are monocytes and CD4 T cells, CD8 T cells, and B cells. But there are a lot of things going on in the tissue that are not reflected in the blood. So, for example, macrophage populations are essentially absent from the blood, and a lot of our genetics have pointed to interesting things in T cells. But it turns out that probably the most important T cells are T cells that we see maybe mostly in the tissue.
So it's not only the stromal component, but the immune component.
Even the immune component, right.
In the tissue, right?
That's right. So some of the key populations that we think are the essential ones to pay attention to, they're not really in the blood, unless you really know what you're looking for. Whereas in the tissue, they're sitting there and they're very obvious. And then, of course, there's the question of the stromal component. When I was a graduate student, I sort of thought of tissue structure, the stromal component, the fibroblasts, the endothelial cells, as sort of the bricks in a building where a big fight was going on with the immune system, and that was how autoimmunity works. But actually, they're not bricks; they're active participants. In rheumatoid arthritis, some of the key cytokines actually come from fibroblasts, those fibroblasts actually interact with T cells and other immune cells, and are part of the feedback loop that keeps inflammation going. And what we're coming to realize in the context of rheumatoid arthritis are patients who we are doing the least well with, the patients for whom our therapies aren't working. They're the ones where their fibroblasts are playing a really active role, because our therapies really focus on the immune cells, but they don't focus on the immune activity and the stromal cells. So that's again a little bit of a surprise to me as we've gone into this field.
There's a lot of opportunity in the stromal component that you're basically taking advantage nowadays that we can access that.
Yeah, that's right. It's obviously, it's a lot easier to convince a patient to give you a blood sample than just to,
a biopsy.
To get any kind of biopsy. But, and of course we have to think about the ethics. So.
That's true. And so, just with the example that you also made, you are combining very different things again. So you are combining different disciplines and different tissue, different samples, and whatever, in a great depth, because you're going to cells, to tissue, and then the disease and the relation already. So with the people having the disease in populations, and so. What advice would you give to people to really being able to doing all these things in so much depth without losing the precisions and track things?
Yeah, it's a really, really tough question. So I think in a way what you're sort of saying is that it's easy to get lost, right?
Yeah, you have a lot of data, a lot of different things to think about, and the human body is not that straightforward.
Yeah, and I think in the era of big data, right? I mean, it's true, right? You can get, lots of amazing genetic data. A lot of amazing things can be done with genetic data. Then you can get a lot of amazing single cell data, and you can do a lot of amazing things with the single cell data. And in fact, you can do a lot of, even productive things with all the different data types you're collecting. But if you're not careful, you can lose sight of the disease that you're trying to solve. I would say, number one, as you're a graduate student, so you're probably grappling with all these things all the time, right? But I think there is a little bit of just judgment. I think. What I always try to, there's maybe two things I think about a lot as we try to decide what to do and how to spend our time. One is our patients, right? So, we can always get caught up with finding some very interesting, molecular phenomenon in our data, or some interesting trends or correlations. But if it's getting too far away from our ultimate goal, we have to balance what's tractable and what's practical and interesting versus are we getting something that's advancing things for our patients. So that's one thing. One thing that we always try to kind of keep in mind. Maybe one story I'll tell you is, we spent a lot of time in our lab thinking about batch correction, which is very important when you collect single cell data. And when we first started collecting single cell data from rheumatoid arthritis patients, we started to see a lot of batch issues. We started to see a lot of batch effect differences between different patient samples. And so then we were confronted with a choice, which was, do we not solve this problem at some algorithmic level? Do we not develop some methods, but try to basically only look at this situation and try to fix this situation? Or do we develop an algorithm that really solves the problem? And so again, we were confronted with, do we do what's the best thing for the disease field as fast as possible, or do we solve a general problem? And in this case, we actually chose to solve the general problem because we felt that this general problem was a very important problem, and one that we needed to solve to do, what we needed to do, but also something other people need to solve. So yeah, maybe I'm not answering the question very well, but I think it is a bit of a judgment call.
No, I mean, it's clear, right? You always have this balance between something more methodological, more for the whole field, less for the people that went into the cohort in that specific study. But then if you don't do that, the next study will face the same hurdle, right?
Exactly.
So,
Exactly.
I mean, I think it's a very interesting dilemma that sometimes we are in, and I'm all for going for the methodological question and try to just like eliminate that hurdle for the next, not only for your group but also for the rest of the groups that will do that, right? It's
I think it's fundamental to have this kind of
methods, and techniques also. Advancing, you don't know what comes out after these developments, but it's always much better and a very important contribution for the whole community.
Yeah, that's true. Also because then not each lab don't have to redo it by scratch, so it's done, and it takes a lot of times and involvement to redo things from scratch sometimes.
But it's a balance, right? Because sometimes you might be finding yourself trying to solve a problem that you find interesting, or you realize that if you do a particular thing, you could get a 2% performance gain. And then at that point, you have to say, is this really advancing the field or not? And so we're always struggling with this in our own group ourselves. I also think another place we sometimes get stuck is, sometimes, the blood and tissue example was a really nice thing that you talked about. Like, I think as, living in the immunology space, a lot of times we think there's a ton of useful information in the blood, and it scales well, it's easy to collect. The decision to go to tissue is a big decision. So, but sometimes I think we try to look for the gaps in the data that we have, because you've got, 6 of the key puzzle pieces, but there's this one missing piece. And I think one of the things we struggle with as a kind of data-driven group that's good at analyzing data is making the decision to try to go for that 7th piece. Because for us personally, that's hard. But there are situations where you say, actually, without the tissue, we're not seeing what we need to see. So if we don't have that piece,
it's circular, right?
We're circling, right? So sometimes then you have to make this choice to do something hard, which in science, you guys are in this business, right? So we're always under pressure to produce. We're always under pressure to put out papers and to stay, active and productive. And sometimes the decision to go for a new data type, you're risking that. So that's always something we try to stay clear-headed about.
And going along this line of making real progress, from your perspective, what will be the ideal scientific environment that's needed nowadays in order to actually make real progress on diseases patient, and in particular in your case in complex immune-mediated diseases?
Yeah, I think over the last several decades, I think we've really benefited from having an interdisciplinary environment, and I think this is really changing. For you guys, maybe this doesn't seem like a strange thing to say, but when I started in graduate school, you would join a cell biology department or an immunology department, and then you would train in that discipline and know that discipline really well. I think these days it's become very important to have cross-disciplinary people. It's really important to have people who are, experts in statistical genetics working with people who really understand the immune system. And it's, important to have some people who understand AI in there, and also important to have people who understand cell biology, so that when you collect your, spatial transcriptomic data, they can help you understand it. So I think that's, I think when we think about building our teams, we have to think about, bringing together people who have all kinds of different talents, to work together. And it's a bit of a challenge to have a team with people with multiple different kinds of experiences and expertise, because you have to make sure that they're learning from each other, talking to each other, listening to each other, and producing together.
So in this sense, if you were to advise something to, let's say, a very young person that is finishing high school and thinking about a degree, right? When we think about the future and we see all of this need for shared knowledge about many different fields, right? Because when I studied molecular biology, you go that alley, that's your thing, you learn a lot about it. Statistics, human genetic statistics, a completely different alley. But if I was a student in high school, right, much younger than I am, what would be your recommendation to study as I go into let's say, into college or university, if I was interested in medical or biomedical research nowadays, right? What would be a track that you would recommend, or a series of tracks, you know?
Yeah. I mean, like, I think the answer is slightly different for each different person, because different people have different interests and different talents. So I think the first thing I tell young people is find out what it is that what drives you, right? And pay attention to that, right? So just because AI is really hot, but you love looking at, but what you really love doing is thinking about biochemical structures. Don't ignore that, because people are usually good at the things that they're most interested in, and it's usually a bad idea to pursue something you're maybe not interested in, but is somehow practical. Because in the end, whatever you're interested in will feed into this interdisciplinary culture. The other thing I think that's important is learn all the basics. Don't ignore math because you find it to be something that's a little bit boring, because math will become important. Don't ignore learning to program computers, because that's not going away. Learn molecular biology. Learn the patient-facing stuff. I think it's also very important for people, if they can, to meet patients or learn about the actual disease that they're trying to go after. So I think having all your kind of bases covered like that is valuable.
It's not easy to get a university degree that covers all of this, because usually very biomedical things will be lacking in math. Very mathematical engineering type of computation engineering will lack obviously all of the biomedical molecular biology, and let alone the patient dealing with patients, which actually is something that we are missing also.
Well, so I think you have to balance traditional university experience with this breadth of experience, right? And I think you're right. I think, obviously universities are special places, and getting a depth of knowledge in a particular domain is what universities excel at, right? Part of getting a PhD is exactly this. But I think in the future, being able to, balance specialization with generalizability is going to become important.